conclusion was that we had no idea what the hell was healthy, because we would sample these guys, they would look healthy, and then they'd develop the disease a few days later, or so we just really didn't know. And so a lot of ongoing work. Ian just got a grant to study this, and we're going to keep working with it. But I think it's really important to keep in mind that healthy baseline. If you see a shrimp with white spot syndrome virus, it looks weird, right? Like, okay, that's a shrimp that doesn't look good. But if you go out and you collect healthy shrimp, or you even get them from like a bait shop, which is what we often do, we can find new viruses in them all the time. So we went to masteries, we got a bunch of bait shrimp, we did our method, we purified the viruses, we sequenced them, we found brand new viruses, right? And so, are these viruses causing problems? Maybe. Do you know if a shrimp is like sick? If it's not showing visible symptoms, it's kind of tricky. Or maybe these are things that are just out there in the healthy population that we need to know about. We have also had the reverse situation a lot of times. So the same method that we use for looking in disease samples, we can go into just seawater samples and say, okay, we know there's a lot of viruses floating out there in seawater. What are they? And we can sequence their full genomes. We can say, oh, we have these really interesting virus genomes. They've never been seen before. But we don't know what they infect. Right? So this is a really weird question, and it's where you start to get into like, well, why does this all matter, right? For ecology, for oceanography, they're viral genomes, so what? And we've been in this situation a lot now with these small single-stranded DNA viruses. It turns out our methods are actually selecting for these guys, which we didn't know in the beginning. But now that we know how to get them, we've become really interested in them. These are super small, so 2 genes, that's it. One gene encodes a replication protein, which is how the virus replicates. The other one is its capsid protein. That's it. That's all they have. They can go in and wipe out animals. The things about finding these in the oceans was that until about 5 years ago, we'd never seen them before. And the ones that they're most closely related to actually infect pigs and birds. Obviously, neither one of those is dominant marine organisms. And so we really had no idea what they were doing in the oceans. And so we kind of started thinking a lot about, well, what are the dominant animals that are out there? And the dominant animals, as you'll learn in BioOcean if you haven't already, are not those big, charismatic, fish that you saw Amy Wallace carrying around, but instead are the really, really tiny animals. And so we started thinking about zooplankton. Now, zooplankton are super important in the oceans because they're a really important link between the low trophic levels and the higher trophic levels. And we really don't know very much about, in fact, a few years ago, we knew nothing about viral infection in these organisms. And so there's obviously a lot of different types of zooplankton. I'm not a planktologist, what's that word? I'm not one of those people, and so I don't know that much about them, but I can identify that that is a copepod. And since copepods were easy to identify and we could get a lot of them, we started working with them. Turns out they're also very important. So these are the most abundant mesozooplankton. There's more than 500 million metric tons in global dry biomass. They're really important in marine food webs all over the place. They're also important for aquaculture because they use them as a food source in fisheries. And there have been a lot of studies to look at their mortality. And originally, people thought the only way they died basically was being eaten by something bigger, so consumptive mortality. But if you just factor that in, it turns out that about 35% of mortality is not accounted for. So we wanted to look into these guys for viral infection. Sort of long story short, a master's student who has now graduated from my lab, Darren Dunlap, looked into 2 different species of copepods, Lepidoptera estiva and Acarcia tonsa, was able to find viruses that were related to the ones that we had no known host for in each of these species. These were the first viruses of this kind that we knew in the marine environment. So we get a lot of firsts in my field, which I think is really exciting. It's kind of one of the things that keeps me going, and it's be easier in our field than in others because we have a shorter history of marine virology. These were also the first viruses found in any mesozooplankton. They had really high viral loads. So copepods are tiny, and we would see 10 to the 5th to 10 to the 6th viral copies per copepod, per individual. And in some populations that we looked at, we could see up to 100% prevalence. We could tell that these viruses were actively replicating and actively transcribing. What we don't know right now is the ecological impact of this. So this again gets down to like, how do you know if your copepod's not feeling very well today? So these were copepods that looked good. I mean, we collected them. They were actively swimming. They didn't die on our watch. But in terms of what is the role of viral infection, how's that going to affect carbon nutrient cycles? Is an infected copepod any more likely to be eaten or to sink out, or various things are things that we're still looking on. We also did look within the copepod tissues, because lots of people are more visual people and they like to see it. And what we see are actually really high loads of viral particles in some of the tissues. We've now managed to convince ourselves, and thankfully the National Science Foundation, that this group of viruses that we didn't know at all before in any invertebrates is really widespread in marine as well as terrestrial inverts. So we have a big project called the Assembling Tree of Life project right now that several people in my lab are working on to keep hunting for these groups of viruses. And this is the most complicated slide that I'm going to show you, but all I really want you to know is that a lot of it is blue, like the oceans. And what blue means in our world is really low percent identities in that most conserved gene, which is the only one we can compare. So pretty much every time we're sequencing new viruses from invertebrates, everything that we're finding is pretty new. So this square here with kind of the warmer colors and the higher identities are the ones that were known before we started our project. And these ones are all the new ones that we've found in the last 2 to 3 years. And we're not identifying close clusters yet, right? So this seems to be a viral group that invaded a long time ago and has really diversified. We know that it evolves very quickly. And getting at the ecology is really the next step that I think we're getting close at. I have no idea when I started. All right, just going to go through the rest of my lab really quickly. For new students so that you know faces and you know who they are. Erin is a PhD student. She's currently in Costa Rica on a Fulbright fellowship working on fecal pollution. Karina Rosario is out on maternity leave right now, but you'll see her around in about a month. And she's working on the vector metagenomics, specifically in whiteflies and looking at plant viruses. Chelsea, who's in the back of the room, is co-advised in Kristin Buck's lab, and she's looking at interactions between marine viruses and metal. I just told you one little part of the lab today, because otherwise I'd go on for a whole lunch break. Stephanie Lawler is co-advised by Chris Kellogg at the USGS, looking at bacterial associates on deep sea corals. I have a new PhD student, Natalie, who's back there, and a new postdoc, Annie, who are going to be starting to work on looking at bacterial diversity within a marine biodiversity observation network that we're working on with Frank Mueller-Karger's group. Dawn Goldsmith, who recently earned her PhD and is a postdoc in my lab working on marine viruses. Another new student, Amanda, who's working on squid diversity with Heather Judkins. And 2 more PhD students that are co-advised by, you see why I'm so busy, by Laurie DeShaw over at USF Health, looking at bacteria and phage interactions within the guts of marine tunicates. We also have a horde of interns that are doing the viral discovery projects. And I'm really passionate about outreach with girls in science. So we've done a lot of projects with the local Girl Scouts. We've also built and equipped a Girl Scout camp, a marine science lab at the local Girl Scout camp. So if you're ever interested in working with groups out there, please just let me know. We have a lot of stuff out there. We've had these kids do everything from collect inverts for our viral discovery stuff, from crabs in the ocean. We've had them do bugs. We actually named a new cockroach virus after a Girl Scout troop 2 years ago. And we're doing everything. We're teaching them about insect vectors. We're showing them really cool new microscopes that work on your iPads. And I absolutely love that. So that is all I have, and I'm happy to take questions. With my pipe.